I never would have imagined that I would be hiking through the forest catching lizards for my PhD. Growing up in New York, I had never even been hiking, let alone caught a lizard. But there was something very specific and microscopic reason why I chose to do this. These wild lizards have something very interesting in their blood: malaria parasites. Malaria is often discussed in the context of humans as it is one of the most, historically one of the most severe parasitic diseases. It is still responsible for over 500,000 deaths worldwide every year, most of which are children under the age of 5. What you may not have heard, though, is that malaria parasites can infect a range of different animals, including primates, hooved animals like deer, birds, and yes, even lizards. While there are 5 different types of malaria parasites that can infect humans, there are actually over 100 different malaria parasites that can infect lizards. Despite this vast number of parasites, we know relatively little about lizard-infected malaria. For my dissertation, I chose to research 3 parasites that infect wild lizards. These parasites infect the blood cells of these wild lizards, so I had to travel to the field to collect lizard blood samples. More specifically, I went to a small volcanic island in the Caribbean known as Saba. As you may imagine, catching lizards is actually quite difficult, but that was only the beginning of the process. After spending days locating and catching lizards in a real-life I Spy game, we had to collect small drops of blood from their pinky toes to analyze for infection status. We took very small amounts of blood so that we could return the lizards back to where they came from so as to be as least invasive as possible. I then analyzed these blood samples here in New York using various methods, including microscopy. The parasites indicated by the arrows on the slide live inside the blood cells of the lizard, so I had to look at hundreds of blood cells per lizard to determine if the lizard was infected or not. We can only learn so much from microscopy, however, so I spent a lot of my PhD generating genetic data for these parasites. This is the first time this type of data has ever been collected for these parasites, so I decided to focus my research on 2 key aspects of the parasite's life. 1, how do the parasites enter and leave the host blood cell? And 2, once they're inside the host blood cell, how do they digest or eat the host proteins that are present for nutrient acquisition? Despite infecting a very different type of host, I found that these lizard malaria parasites both have and use some of the same genes as their human infected relative. This is important because genes that are conserved across both human and animal malarial parasites are likely essential for the survival of the parasite. Discovering essential genes is key for future antimalarial drug treatment and discovery. If you can block the essential gene, you can kill the parasite and treat the patient. The data I generated, therefore, can be used for many comparative analyses by other scientists in the future. Who knew you could learn so much from just a few drops of lizard blood? Thank you.